that ALK Positive is doing, that we're doing. I wanted you to check out my shirt. If you can see Mark's shirt too, he's got the Lorlatinib molecule there, but you can get those shirts by the store that's managed by our very own Erica Lovack at ALKpositive.org. Just hit shop. And last weekend, you heard about some amazing research. Tonight, you're going to hear about some more amazing research thanks to these doctors and our research and review panel who chose this research. If you want to help to fund this research, you can do it at ALKpositive.org. Just hit donate. And if you want to delegate your funds anywhere, just let us know. Just tell if you want to know where your fundraising is going, just let us know and we'll make sure that you're informed about that. I wanted to tell you about one more exciting thing. In May, we're going to have an ALK Walk Positive worldwide, and it's inspired by our own Niels Harthoorn. If you see him in the Netherlands, he actually did a walk back in August, and he raised quite a bit of money for research, this research that you're about to find out about. So it's really exciting. In May, we're going to do that. So keep your ears peeled for more information about our ALK Walk Positive worldwide. So I'm going to give it back to Mark, and he's going to ask you guys to look at your screen, and he's going to ask you a few questions.
Okay, you see the first question up, which is, what type of treatment are you currently taking? Which pretty much probably will know the polling, but you never know. Okay, I'm going to end the poll and share the results. So Alectinib is 64%, Lorlatinib is 19%, and then 8% is Actinib, and then we have a few stragglers. So I'm going to relaunch another poll. Oops. Sorry, you think I'd be better at this. Length of diagnosis. Okay, I'm in this poll. So 38% is 1 to 3 years, 1 year and under is 20%, and then 5 and 8 years is 13%. And we have 7% of the people are 8 years and older. And the reason I'm saying this out loud is for the recording. So the last question right now is, how many TKIs have you taken? And if you've taken any chemo or immunotherapy, you do multiple, more than one choice. And we are being recorded, so do not talk or you will show up on the video. Okay, I'm gonna end the polling and share the results. And no surprises, 1 TKI is 51%, 2 TKIs is 31%, and then we had chemo, 28% of us have taken chemo before, and 3 TKIs is 10%. So I'm gonna stop sharing and turn it over to Emily. So Emily, it's all yours.
All right, thanks, Mark. And I'm very happy to introduce our 2 guests tonight, Dr. Ibiayi Dagogo-Jack and Dr. Jessica Lin. And they work together. They're both attending physicians at Massachusetts General Hospital in the Division of Thoracic Oncology. So they are lung cancer specialists, and they are both instructors of medicine at Harvard Medical School. And we're really thankful that they're here tonight, spending their Sunday evening with us and sharing about a new clinical trial combining Lorlatinib with other inhibitors, partially funded by our ALK Positive research. And if you have questions for them, please type them in the chat, and please keep your chat comments only to the questions right now for Dr. Lin and Dr. Dagogo-Jack. We'll have some time after our Q&A with them to talk amongst ourselves like we usually do at these ALK Talks. But for now, please keep your comments and questions on the topic. And I think Dr. Jagogo-Jack will be leading us off. She has a few slides to present to give us some background information about the trial.
Perfect. Thank you, Emily. And thank you, Gina and Mark, for having me. It's a pleasure to be here tonight. And I think that it's very, as Emily said, it's very fitting to be here because this trial that I'm discussing, we couldn't have the trial without the funding and without the support from patients, because this trial is really for you guys and about you guys. And so we're always seeking feedback. And because this is a somewhat complex process, kind of combination, I thought it would be helpful, because we're all coming at it from different backgrounds, to discuss why we're even pursuing combinations in ALK positive lung cancer, and also thinking about why we're interested or excited about this particular combination. And so this slide is initially looking at, it basically represents the culmination of our experience here at Mass General and across other sites of studying and really analyzing biopsies from disease sites that have started to grow after treatment with an ALK targeted therapy. The first pie chart is after second generation ALK-TKI. So that would be Alectinib, Brigatinib, and Ceritinib. And the second pie chart is after Lorlatinib. And so what the first pie chart shows us is that most tumors, when they become resistant to Alectinib or another second generation ALK-TKI, will have ALK-dependent resistance. So we think of this in 50 to 60% of tumors. So that means about 40% of tumors are growing because they're getting signals from other things other than ALK. We call that ALK-independent resistance. We recently described that about 15% will have extra copies of a growth signal called MET, but that means that the remaining tumors could have many different resistance mechanisms or many different growth signals. It's important to sort out ALK-dependent versus ALK-independent resistance, because we find that if it's ALK-dependent resistance, meaning an ALK mutation is now present, most of these tumors will go on to shrink with Lorlatinib. After Lorlatinib, things are a little bit different in that Lorlatinib is a more potent ALK inhibitor than our second generation ALK inhibitors. So what we end up seeing is that there's a bit of a shift. And so ALK kinase domain mutations or ALK-dependent resistance is something we see now in about a third of cases, a third of tumors. And so 2/3 of resistance to Lorlatinib is caused by ALK-independent mechanisms. The best way to try to overcome an ALK-independent mechanism is not to continue another ALK-TKI, but to find a combination that can block those extra growth signals. And so this is a somewhat complicated, but biologically a little bit oversimplified graph to try to kind of illustrate again the point about why in the setting of a bypass track, meaning an extra kind of recruited growth signal, just giving another ALK-TKI or another ALK pill might not work. And so when we say that a cancer has an ALK rearrangement, that is a powerful growth signal, but that isn't the only growth signal that causes the cancer to grow. Right? The ALK protein or the excess ALK signal actually talks to a bunch of different proteins downstream, and together they coordinate, kind of letting the cancer cell proliferate and survive. There's 3 key pathways that we think about. One is called PI3 kinase in green, the other is the JAK-STAT in pinkish purple, and the one we think of as the most important one is really the MAPK or RAS pathway. And if you have a tumor that is sensitive to an ALK pill, meaning that it hasn't developed resistance yet, if you are able to kind of, with this stop sign here, if you're able to block the ALK protein, that's enough to block all 3 of these downstream pathways. But when you recruit a different growth signal, and we're still figuring out what these bypass tracks are, but across studies, there've been a variety that have been described. And I've listed just a few here. So these are other growth proteins in addition to ALK that are helping the cancer cells grow. These are EGFR, FGFR, HER2, and MET. A given tumor can have multiple of these, and across patients, different ones can be contributing to the growth of the cancer cell. So if you have a bypass path or an ALK-independent mechanism, it's not enough to shut down ALK, because these can feed these kind of downstream signaling pathways even if you block ALK. Interestingly, when we look at it in the laboratory, it seems that even for these bypass pathways, that RAS-MAPK pathway is the most important one to shut down. And the entry point to that pathway is this yellow protein here called RAS. And for RAS to turn on and signal, it needs a bunch of friends, basically, and one of which is SHP2. And so if you block SHP2, which is what we're doing in this clinical trial, you can block RAS from being turned on. So even if there's extra signals from these bypass pathways up here, the RAS isn't turned on enough to help the cells grow. And so that is what we're exploring in this clinical trial. And so to take a step back, right? So one approach, if you have a bypass pathway, is to give an ALK pill and directly target whichever one of these is causing growth, EGFR, FGFR, HER2. But we don't always know which one of them is causing the growth. And our tests that we use in the clinic, next generation sequencing, tumor genotyping, molecular profiling, doesn't always give us the answer. And so the rationale for this trial is to pick a protein that is basically important for all of these pathways and shut it down with the hopes that we can basically benefit a broader number of tumors than trying to just pick out one growth factor or one bypass pathway. And so some of you may be familiar with our ongoing Lorlatinib combination study. And so this SHP2 arm, or the Lorlatinib plus TNO155, which is a SHP2 inhibitor, is one of the treatments that's part of this protocol. The protocol is currently open and is already kind of evaluating Lorlatinib in combination with crizotinib, which is a ALK pill that you know of, but also a MET targeting pill, and then binimetinib, which targets a different protein in that pathway that we didn't discuss. And so this is a new arm with Lorlatinib plus TNO155 that we hope to start enrolling people and recruiting to in the next few weeks. The study is currently in dose escalation, or phase 1. And what that means is we're still trying to figure out if it's safe to give the combination and what is the best dose of the combination. And to figure out who this works best for, we're looking at blood and looking at tissue so we can appropriately select people in the future. Once we figure out the right doses, we're actually going to go to phase 2, or dose expansion. I highlight this because in dose expansion, this particular combination is mainly developed for people who've already had Lorlatinib. But in the initial, when we're trying to figure out the dose, everyone can be eligible for this trial virtually, essentially. And the last point I wanted to highlight, I am happy to answer any questions you may have, but I put this up here just to say that in general, if for those of you, I think about 20% were on Lorlatinib, most of you know that the starting dose of Lorlatinib is 100 milligrams. In this study, we're actually going at half of the dose because we don't know how these drugs are going to interact with each other. And so we intend to escalate, but this is part of a clinical trial. So you start with a dose and then you figure out the best way it works and then kind of escalate. And so that's our goal. I also want to highlight that these are pills. And so it's an oral treatment throughout. And so that was just a bit of background about why we're all excited about targeting SHP2. And again, to emphasize, the potential is really to block a whole bunch of different growth signals in a kind of a broad way, rather than having to have the insight into which one is causing resistance in an individual tumor. And this trial, we hope, will start kind of enrolling people in the next 2 to 3 weeks.
Thank you, Dr. Dagogo-Jack. We have some questions coming in already about qualifying for the trial, whether, what are some of the qualifications? Do you have to have a measurable lesion? Could you go on to this trial after you've been on chemo? What are kind of the list of qualifications for this trial?
Yeah, I think these are very important questions. And so the trial is developed to be kind of irrespective of your chemo history. You can have chemo before you've been on the trial or after the trial. So all comers are welcome, regardless of what your chemo history is. As far as the measurable disease, it is ideal to get some insight into not just how safe something is and how well tolerated it is, but also how well it works at shrinking tumors. So it is currently, for the most part, developed for people who have measurable disease, where we can really assess the response. But we also have make some exceptions that as long as there's some place that we can gauge response while we're still figuring out the right dose of the medicine, we don't technically have to have multiple disease sites. We just need at least one area that we can look at to evaluate response. But that will be different when we get into the expansion, where we're really looking to see how effective it is. Then we will truly need measurable disease.
Okay. And Someone would like to know whether there have been preliminary mouse studies before this trial.
Yeah, so there have. So there was a slide in here that I took out because it was just really dialing down too deeply into the data. But the mouse studies are what the mouse studies and the cell line studies are what make us excited about this. And so to give a little kind of just a simple description of the mouse studies and the cell line studies, there was an experiment that was led by one of our postdocs here where they took cell lines that were derived or generated from biopsies of areas that had developed resistance to ALK inhibitors, and showed that if you just give an ALK inhibitor, those cell lines continue to grow. But if you started kind of selectively blocking certain proteins like EGFR, FGFR, and other proteins, depending on which one was causing resistance in the tumor, you could make the cell line sensitive again to treatment. But if you looked across like 10 patients, it was so different. Like within a single cell line, there are many different growth signals that were causing it. So you had to knock out 1, 2, 3, and those 1, 2, and 3 proteins may not be the same for every patient. But what was important was that when you knocked out SHP2, that was effective in the majority of cancer cells, so around 50 to 60 of the cancer cells. Again, it's a small sample size, but that was encouraging to see that it might be effective in across tumors that are listening to multiple different growth signals.
Okay. Someone else would like to know if you can compare a MEK inhibitor versus a SHP2 inhibitor. Do you think one is more promising than the other? Does it depend on the patient? Any comments on those two different drugs?
Yeah, I think it's a very insightful question. And so they are both working in some ways in that RAS-MAPK pathway. But when we look at the data in other tumor types, so a different type of lung cancer that has a KRAS mutation and other kind of mutations, it seems that blocking SHP2 is more effective in some tumors that may not respond to the MEK inhibition just because it basically cuts off multiple different growth signals in a way that MEK doesn't necessarily do. We do think that some of the side effects may be similar because you're really targeting one pathway. And so the side effects that we've become accustomed to seeing with this type of targeting of that MAPK pathway are some gastrointestinal side effects that people may have, nausea, diarrhea. Not everyone has it, but it's something that we maybe see a little bit more with the SHP2 inhibitor than a MEK inhibitor. We can also see that some effects on the heart. And so we monitor the heart very closely during treatment with either of these drugs. And usually that effect is reversible, but it can cause the changes in the way your heart functions and squeezes. They can also cause some changes in your eyes because this MEK protein is expressed in the eyes. And so we monitor the eyes very closely. That eye change is actually more common with the MEK protein than the SHP2. So there are some similarities, but also differences. And we're still figuring out.
Okay, thank you. How about brain penetration? Are these drugs, particularly the SHP2 drug, are they brain penetrant? Will they work or potentially be effective if you have brain mets?
Yeah, we're still learning. We're still figuring that out. For the MEK inhibitor, some of the earlier, so by the time you get to the drug that's in the clinic, in general, there have been multiple iterations of the drug where they've tested different compounds and tested it in mice and other animals. And targeting MEK, it turns out in the brain was detrimental. It caused a lot of side effects. Mm-hmm. So a lot of MEK inhibitors are made on purpose to not get into the brain. And the SHP2 inhibitor, we're still learning if that's the case with it. But this study, since we don't know that it works very well in the brain, this study does require that if the brain is progressing, in particular, if you're having symptoms from the brain progression, that those have to be treated first, especially if they've started to grow on a powerful kind of drug that can get into the brain like Lorlatinib.
Okay, good. That covers another one of the questions. How many people are you accepting into the trial?
Yeah, I think that's a very important question, and it gets at a relevant point. So when you're in a dose escalation study, you actually are limited in some way in how quickly you can put people on a study. So each group, we basically enroll people in groups of 3 and make sure they tolerate it before we enroll more people. And so in that dose escalation phase, where we're still finding out the right dose, we expect to enroll about 12 to 15 people, but that may be staggered over kind of 2 to 3 months as we get sets of 3. In the bigger study, basically in the expansion, we envision enrolling about 25 people, but we want to make sure it's the safe thing to do and the right thing to do. So we're going to pause after the first 10 people have been on and make sure that it's really working before we put an additional 15 people on.
Okay. Would you suggest this study if a patient has had a lot of side effects on lorlatinib alone? Would you suggest this for them or not?
Yeah, I think it's tough. So one of the eligibility criteria is that you have to have tolerated lorlatinib at the dose that the study is exploring. And so if you, so that means you have to have at least tolerated lorlatinib at half dosing at 50 milligrams. And the reason I said it's tough is that this drug may introduce even more side effects, that the combination may introduce different side effects than you had with lorlatinib alone, or it may enhance some lorlatinib effects. In particular, edema or swelling in the legs is something we see with lorlatinib that's also seen with the SHP2 inhibitor by itself. And so I think it has to be a very kind of discussion between you and the doctor about kind of what side effects you're having. We certainly don't expect some of the neurocognitive things like slower speech, trouble processing to get worse with the TNO155, but there are certain side effects that may be magnified when you combine the drugs.
If you were struggling with, for example, the lowest possible dose of lorlatinib, we certainly wouldn't expect the combination to be easier per se, right? So that should give you kind of an inkling of whether the combination trial would be a good option for you if you had found the lorlatinib monotherapy to be that challenging, where you required the reduction to the lowest dose and still the side effects were debilitating.
How do you decide, or how will you decide which arm of the ones that you mentioned to place a patient on? Especially, this person's asking, especially if there are no submutations found, either because it's difficult to get a biopsy or nothing turns up in the biopsy analysis.
I think that's a very good question when you have a study with multiple arms. Are we going to randomize? How are we going to pick who gets on what arm? For the MET amplification arm, that is a biomarker selected arm, meaning you should have MET amplification to go on that arm. For the others, we're basically doing it based on slot availability and recognizing that some people their tumors may be more likely to respond to maybe a SHP2 inhibitor than a MEK inhibitor. We haven't actually limited the ability for you to cross over. So if you had a SHP2 inhibitor and it didn't work, you could still always go after that point to the MEK inhibitor combination or vice versa. But I think it's really going to come down to the slot availability.
Okay. If a patient crossed over from one arm to another, would they kind of count as filling another slot in the trial, or?
They would, but we've also created some leeway in the study for what we call backfilling. So recognizing that for some people, there aren't a lot of viable therapies left, and this might be something that we believe in or someone is very excited about. And so if we know that a dose is safe, we do have in rare circumstances the ability to put someone in at a dose level that's already been explored.
Okay. Is the study only going to be offered at Mass General or other locations?
It is only at Mass General for the time being, yes.
Is there any possibility to do a remote participation in this age of telemedicine?
Yeah, so I'd qualify it. It is only in Boston, is how I would say. It's at Mass General, but also Beth Israel will be participating, and the Dana-Farber might as well. We are trying as much as possible to do what we can remotely, but we also recognize that this is a new combination, and so we have to make sure we're keeping a close eye so we can be vigilant and capture any kind of side effects and address them and manage them quickly. And so it does require you to physically come here for the study. Hopefully, if someone is on the treatment for a long period of time, there's flexibility then to space out the visits and also do some remote visits.
Okay, thanks. Has the SHP2 inhibitor been used in other drug combinations yet? And, for other cancers? And if so, are there any results from those trials yet?
Yeah, so it's a very good question. And that's the reason that when I showed you the slide initially, I said that we have the combo study already open and this one is coming. We always had the idea of putting this one as part of it. but it actually is a brand new drug, whereas the other drugs are approved for other indications. And so for the brand new drugs, they actually have to go through testing by themselves first. And so that had happened up until this point. The reason we're cleared to open it now in combination with Lorlatinib is that the company is also doing a study in parallel in combination with an EGFR inhibitor at this dose. And they had seen that it was safe to combine it at the dose that we're starting with Lorlatinib. It's too early to tell if it's more effective, but at least similar to with the ALK experience, in the lab, it seems that combining it with an EGFR pill seems to be more effective than the EGFR pill by itself.
Okay. And someone would like to know which company makes the TNO 155?
Novartis.
Okay. Colin, have I missed any questions? Oh, Colin would like to know if the EGFR combination trial has already started.
It has, to my knowledge, yes.
Okay.
And there will be combination, with KRAS inhibitors too.
Great. So I know that you said you don't know whether it's been whether it's going to be brain penetrant or not, but just based on what you've seen from molecular structure and so on, do you suspect that it probably is, or suspect that it's probably not?
I would say the latter. I would suspect that it's probably not going to penetrate the brain significantly.
Okay.
And so, Dr. Dagogo-Jack, do you want to comment on with regard to that eligibility with regards to brain metastasis and prior Lorlatinib?
Yeah, so if you have had prior treatment with Lorlatinib or a brain penetrant ALK inhibitor, here we mainly mean Lorlatinib, and the cancer has grown in the brain in that setting, you actually have to have the brain metastasis treated beforehand, before going on, just because we don't want to put you on a medicine that we don't know can get into the brain and risk the brain metastasis progressing and causing more symptoms.
We have a question about chemo. I think this is someone who's asking if they're on chemo and doing okay, is it still possible to pause the chemo and go on to this trial instead, thinking that it may be less toxic, you know?
Emily, you just muted yourself.
Sorry.
I guess I get where you're going. So it is possible. It is, but what I always want to emphasize too is that we always, as long as you're tolerating the chemo well, we value getting the most out of every single therapy that we have. And so I wouldn't necessarily interrupt the chemo for this study, because we don't know how you're going to tolerate this medicine in the study or how well it's going to work. And the hope is that the study would still be available after chemo.
Okay.
If I can ask a few questions that I haven't typed in, but I'll just ask them.
Sure.
Assuming that you end up with the best possible results that you could imagine out of the trial, you know, it just looks spectacularly successful. How long would it be before you went into a phase 2 trial, typically in this sort of situation? And then eventually, ultimately, would you see this being used in combination with other ALK and would you eventually, you know, assuming it turned out to be, would you even foresee this being something that would be used in the first line in combination with a TKI?
Yeah, and I think that these are very, very important questions, and it's part of the reason we start with investigator-initiated studies. So Pfizer is very interested in this question as well, as is Novartis. And so this trial is a way to kind of look at it in a smaller population of patients to see how effective it is. And if there's a promising signal, if we're having, hopefully, which is our goal, having very great responses that are lasting a long time, then I think the right strategy, which some of you have already hinted at, is to do a multi-center study where we can enroll many more patients and get a greater sense of how effective it is. And exactly thinking about moving it towards the first line setting.
Great. Does anyone else have any other questions about the trial? If we don't, then we could just start putting in clinical type questions, and these are both fabulous clinical oncologists, and they can't answer specifically to an individual case, but they can answer in a sort of generic way that can be very helpful to lots of people.
I see a couple of questions about these drugs. One being, can they be prescribed kind of off the trial on a compassionate use basis or an off-label basis if a doctor thinks that it's a promising option for them?
Yeah, I think that there's one I saw in the chat now about lorlatinib, crizotinib. That one usually can, because they're both approved drugs and they're both approved for ALK. And so a lot of the time, the determinant of these off-label or compassionate use or single patient protocols is insurance, whether it'll cover it. And for newer investigational therapies, you often have to go through the company and get kind of regulatory permission from the FDA, et cetera. And so I think something like prescribing TNO155 that's not approved will be quite difficult, but something like binimetinib might be, in theory, possible in a compassionate use basis. But again, I envision that the pharmaceutical company would want at least some preliminary data from this study to guide or support that. As would I, as the prescriber. I would want to make sure that whatever I was pursuing was helpful for the patient. But crizotinib, I have had people get prescribed lorlatinib and crizotinib outside of the trial.
Okay.
So there is one question here from a member. How often would a visit to Boston be required to participate in the trial?
Yeah, so initially, a lot of these studies, we always start with closer monitoring and then we space out. In the initial period, one would have to expect to be here at least every 2 weeks for the first 6 weeks, and then it does space out. Right now, because the way TNO155 is dosed, it's basically, it's a 3-week process for dosing, and you take it 2 weeks in a row and then 1 week off. It shortens the cycles a little bit. We always like kind of closer to a month cycle to give you more time away, but this one makes it a 3-week cycle. So you would at least have to have a visit every 3 weeks for monitoring. The goal, though, as we've done with these investigator-initiated trials, is that as we get more confident, we get 6-9 months out and show that it's working and you're tolerating it well, is to actually put amendments into the clinical trial to make it such that you don't have to come as frequently once we've established that you tolerate it well and your body's responding and the cancer is responding.
And would you be wanting the scans and so on to be done at your hospital, or would they be able to be done at the local, wherever the patient's at?
Yeah, they can be done locally. So anywhere, and just we would coordinate with our team. We're well versed in doing that, just getting the disc over and formally reviewed internally here as well.
Excellent.
One more trial related question. If you see that the combinations are tolerated well, so lorlatinib and SHP2 and lorlatinib and MET or MEC, would you ever consider doing a 3-drug combination? I know there have been some lab data on these types of combinations. Is it ever anything you would think about or that you think is promising?
I would say never, but I want to prove that they're tolerable first.
Okay.
There's another question here from a patient, and hopefully this doesn't apply to many patients in our group, but this one obviously. And the question is, and I'll just read it out literally, if you have a co-occurring cancer diagnosis, does that impact eligibility for the trial? I have been treated for CLL and am now MRD negative and off treatment.
So it depends on the type of co-occurring diagnosis. So usually we say for most solid tumors, not to have had a aggressive cancer within 3 years that require treatment. So we usually exclude things like pre-invasive, kind of very superficial bladder cancers, skin cancers like squamous cells and melanomas that have been removed. And we are having increasing flexibility around hematologic malignancies like CLL. Because it's easy. So the reason that people make these eligibility exclusions is really you want to be able to tell if something is growing, if it is your lung cancer or another cancer. It's easier to tease that out to some degree with CLL that's an MRD state. So that can be easier to enroll as a concurrent diagnosis.
Would an international patient be able to participate in the trial if they're able to travel to Boston for all the required visits?
Dr. Lin is nodding her head and the answer is yes. We've had
so You know, anytime greater distance is involved, it does make things much more challenging, and logistically takes a lot of thought and discussion in terms of travel, covering the costs because you don't have insurance. But if those hurdles can be overcome, absolutely. If
Okay.
we think this trial is a good option for you, and we think all the kind of challenges outweigh the potential benefit, absolutely. Yeah.
Okay, thanks. And then back to kind of alternative dosing and things like that. If the side effects prove difficult with the combinations, would you consider some kind of alternate cycling? So each drug given separately for a period of time?
Yeah, I would say, and there's also an alternate dosing that's already, I think the advantage of this being an early drug as far as development is that Novartis has also explored alternative dosing schedules. So there's 2 weeks on, 2 weeks off dosing as well. And so that would be what we would pivot to if this kind of 2 weeks on, 1 week off is not working, just an extra week for recovery. And they're already exploring different dosing schedules that hopefully we may be able to use that, leverage that experience.
Okay, great.
Do you know whether there are any SHP2 inhibitors being attempted to be developed that would be more likely to be brain penetrant?
I don't know. I can look into it because I know that there are attempts, although everyone said BRAF and MEK inhibitors were not going to get into the brain. But now here we are 5 years later, and they are developing those that do get into the brain. So I do think that it's possible. And one has to ask why. So it isn't that they can't develop MEK inhibitors to get into the brain, it's that it caused more toxicity when it did. And so maybe they have to just do, looking at Mark's shirt with like a molecule across it, like they have to come up with better molecules or different things that can enhance penetration of the penetrance of the CNS without enhancing toxicity. And it's simple for me to say, but you know, hopefully there are greater minds out there that can come up with this.
Right.
Would a patient be able to participate if they've stopped taking a TKI because of intolerance to that particular TKI? So for instance, if they've developed a liver toxicity or something to one TKI and they haven't switched to another yet, would they be a candidate for the trial?
Yes, so assuming that TKI was not Lorlatinib, you know, sometimes we can see these idiosyncratic toxicities that do not recur with other TKIs. Of course, it raises our threshold for monitoring that much more closely, right? So if we know you've had hepatotoxicity on a prior TKI, we'll probably monitor your LFTs much more vigilantly than we might otherwise, but it would not preclude this trial as an option for you.
Okay. Before we transition to more general clinical questions, I wanted to ask you both a question that I was supposed to ask at the beginning and I forgot. We'd love to hear a little bit from each of you about how you got into oncology and a little of your story leading to where you are now professionally.
I guess I can start since my microphone is on. So I first ended up becoming interested in oncology back in undergraduate years, actually. I started out wanting to become a scientist or a biochemist, but ended up meeting a mentor who was a pediatric oncologist who also did basic research in that arena. And he really inspired me, made me interested in medicine, interested in taking care of patients. Ended up going to medical school. And then, you know, I actually tried my best to really explore other specialties during medical school and residency, trying to convince myself, well, maybe I wanted to go into critical care, pulmonology, but just couldn't find an area where I felt as passionate about or as inspired by than oncology. The patients that I met, the mentors I met along the way, I would say were the two by far most critical factors that influenced my decision.
Jess, I hadn't realized that we had such similar pathways. But what I'll say is that, so Gina knows this, I was talking to her about basically how my family lives in Tennessee. And in Tennessee, there's St. Jude Children's Hospital. And so one of my neighbors actually worked at St. Jude. So when I was in high school and when I was in undergrad, I actually spent time doing lab work and also kind of doing volunteering at St. Jude, which is a pediatric oncology hospital. And that period, it basically, I don't know if you've ever been there, it's a very inspiring place. There's walls plastered with success stories. And so when I went to med school and saw that that wasn't the case, always the case for kind of adult oncology, it inspired me, I think, to want to make that difference. And as Jess said, meeting kind of mentors here, meeting patients that just inspire you on a day-to-day basis, and kind of the research, the science, all of it is what really led me since kind of high school to be on this path.
Great. Well, we're so glad that you chose to help us. And we're so grateful for all the research that you're doing to help out patients. So if I can shift gears now to some clinical questions. We have one asking, if your chemo stops working, have you seen cases where the cancer has kind of been reset by the chemo treatment and then it's possible to return to a TKI treatment?
So we certainly do see instances where if your tumor has not seen an ALK inhibitor for quite some time, then going back on an ALK inhibitor and putting that pressure on the ALK positive cells can reinduce responses. We call it kind of retreatment effect. What we don't know as much about is how long that duration of benefit might last. For example, if you were previously on the same TKI and your cancer had gotten worse, right? And then you came off of it, went on chemo, and then go back to that same TKI, our worry is that the duration of response may be shorter than if you were going on that TKI for the first time. But yes, we have instances where we do see retreatment effect, and particularly also in the with the brain metastasis. Let's say you did have brain metastasis that were responding to the TKI, but then for one reason or another, ended up coming off of it, going on chemo. But then if you go back on a brain active ALK inhibitor, certainly we can see induction of CNS responses.
Okay. What do you think about going on chemo between TKIs, say between Alectinib and Lorlatinib?
I don't think I can say right or wrong, right? You know, we do take into account, as many of you know, at Mass General, we are a proponent of rebiopsies. And so we like to understand why your cancer may be progressing on Alectinib. And if there is an ALK resistance mutation that explains the tumor's resistance to Alectinib, then we would certainly favor Lorlatinib as the next option. Because we know that Lorlatinib can be highly effective if your tumor is known to have an ALK mutation, because it suggests that your tumor is continuing to be ALK dependent. In the absence of an ALK mutation, we know that the efficacy of Lorlatinib is inferior to having an ALK mutation. And so that's where the discussion kind of comes into play between going to Lorlatinib and kind of doing a short interval follow-up versus going straight to chemotherapy. And that ends up being a very complicated discussion, taking into account kind of what your disease burden is, what the pace of change has been, and how you've tolerated TKIs and so forth.
Okay, thanks. Do you have?
Go ahead, Emily.
Do you have any thoughts on the new drug that we have been following on the outpatient board, TPX-0131, which is, I think, a 4th generation ALK inhibitor? Do you expect patients to see it starting in a phase 1 trial this year in the US?
I do. I anticipate towards the end of this year for the United States. It'll be earlier for other countries like Australia. We will open this trial at Mass General. And so, you know, that kind of tells you a little bit about how I feel about it. I am enthusiastic about this novel compound, partly because right now we're completely lacking any ALK inhibitors that could be used for patients after Lorlatinib, right? Lorlatinib ends up being kind of the common endpoint. And then if your tumor is known to have compound ALK mutations where there's 2 or more ALK mutations, we don't really generally have good options to attack that. And this TPX-0131 has pretty good preclinical data that suggests that this drug should be potent against at least a subset of the compound ALK mutations. And so we will open the trial at Mass General probably towards the end of the year, and we're excited about it.
Terrific. That's great. There's a question here from a patient, but she is actually also a medical doctor. And the question is there any way to assess how well patients will tolerate the combination of medications beforehand? For example, would you be able to perform identification of cytochrome P450 genotypes? Do you think such studies might help predict drug tolerance?
I'd say that it's a very insightful question, and it's speaking to the whole field of pharmacogenomics, right? Whether we can predict. And I think that the science is still a little in its infancy as far as our novel combinations, but hopefully there will be ways to do it. The way we get around it now is that we basically put blanket statements about avoiding certain drugs that are known to interact with that particular pathway. And other things that we think might predispose to toxicity, so we keep a close eye on our ocular baseline kind of ophthalmologic conditions that we think may predispose to getting some sort of eye toxicity from the treatments. And then also all of these studies have some sort of language around the heart function that we would look at. And then we also always review your medications to make sure that you're not on medications that also kind of can perturb the heart in ways that this drug can, things like that. But I agree with you. I think it's a direction we need to go in.
Great. I've got a new question just came in, and it says, and this is not related to the trial, for patients at a stage earlier than stage 4, is there any potential harm to staying on a TKI long-term, long after being no evidence of disease, apart from dealing with the side effects?
It's a difficult question, and it is a data-free zone. As most of you know, for stage earlier than stage 4, we have not been using ALK inhibitors as kind of standard of care therapy after definitive surgical resection or chemoradiation. That landscape and the conversation is evolving rapidly in light of the EGFR mutant lung cancer data, looking at adjuvant osimertinib. We do have clinical trials that are ongoing, and Dr. Dagogo-Jack can actually speak more about this, looking at adjuvant alectinib as well as adjuvant crizotinib. If you have been prescribed an ALK inhibitor in the earlier stage setting and are tolerating it really well, what duration of that therapy is really optimal is unknown. In all of these trials that have already been implemented, there was a set duration that the participants received the inhibitor for, whether it's for 1 year or 2 years or 3 years. We do think that once the TKI is removed, then there's a higher likelihood that the tumor cells can proliferate and the tumor can recur. And so I really think at that point, it ends up being a one-on-one kind of personalized discussion, taking into account what your side effects have been, how it's influencing your kind of day-to-day life. And some patients may say, well, you know, I've been on it for 2 years. I really haven't experienced any side effects. My blood counts have been fine. I'm not having any fatigue. I really want to continue because it makes me nervous to think of coming off of it. Then that's a different conversation than if you've had kind of slowly worsening anemia, or you feel like you are getting more easily fatigued, then that changes the conversation.
Great. And then this is my question. As you know, Lorlatinib's just recently been approved for first-line treatment in the US at least. And so would you be routinely prescribing that? As you know, if you got a patient coming in just diagnosed with ALK, would you be putting them straight onto Lorlatinib, or would you be putting them onto Alectinib, or would it be a discussion, or how would that all go?
For me, it would absolutely be a discussion. We are very fortunate in ALK positive lung cancer to have a wonderful kind of advocacy group and support group like this, where everyone has the opportunity to get more familiar with the options and review even a little bit of the data. And I actually find that when I'm meeting, for example, one of you in clinic for the first time, you've already done a ton of reading and have a lot of background already kind of prepared. And so I actually think it would be remiss not to discuss, okay, this is the data for alectinib, this is the data for lorlatinib, and these are the reasons to think about one versus the other. So for me, it would definitely be a discussion. I think the efficacy data for lorlatinib is certainly the best we've seen of all the next generation ALK inhibitors. The curve looking at preventing brain metastasis is just impressive. And we know that brain metastasis can be devastating. On the other hand, as you know, there's a lot of conversation about side effects with lorlatinib, which some of them are quite unique to lorlatinib. And so I do like lorlatinib as a first-line option for those patients where they're very concerned about possible mood or kind of cognitive side effects, for example, because they've had a lot of anxiety at baseline or depression, or have a really high functioning job where they just cannot even imagine tolerating any kind of impairment in that arena, then that would be something to consider in whether alectinib may be a better option. But I was very impressed by the Crown data.
For sure, absolutely. And I'm not sure if I've shared this with you before, but we had an internal poll, nothing very scientific or anything, but we asked members who had been on both at different times, alectinib and lorlatinib, which one they felt better on. And I was truly expecting that it was going to be that they, more people felt better on alectinib, but it's actually the other way around by a ratio of 2 to 1. So
that's very interesting.
Twice as many people feel better on lorlatinib than on alectinib, which totally blew me out of the water. I was not expecting it. It's not a small sample size. It's the last time I looked at that poll, it's about 60 people that had both and answered that poll. So that's fairly good statistics once you start getting to 60 people.
Yeah, and it's so complex, right? We often like to talk about side effects as if they stand on their own, but when you're looking at how you feel on a day-to-day basis, it's not just about side effects, it's also symptoms that are related to your underlying cancer. And so perhaps for those patients where they were experiencing a lot of symptoms from the lung cancer, if lorlatinib was successfully treating that and reducing those symptoms, maybe that also helped them feel better than when they were on alectinib.
Yeah, I get the feeling it wasn't related to the cancer activity. It was just related to the fact we get so many people saying on alectinib they feel fatigued, they've got muscle problems, et cetera, et cetera, et cetera. So if you were diagnosed, I hope you're not, but if you were diagnosed tomorrow, would you start on alectinib?
I think if I had brain mets, yes. That would be my short answer. Yes.
Okay.
And
if you didn't have brain mets?
I think then it's a conversation, right? I think it's tough, right? And I think that it's never a good situation, but it's a good situation that alectinib works so well. And so then the question is always about the information that we don't have about sequencing, whether it's better to take one and go to lorlatinib next. But what we know is that not everyone will have the opportunity to have lorlatinib next anyway. And so how do we kind of measure that and make the right decisions? But I have given lorlatinib in the first line setting already to a patient of mine who had brain metastases and spinal metastasis. And we had a very informed conversation, lasted about 30 minutes. And that was what the patient wanted. And I think that it really has to be on an individual level with these drugs.
Okay. A couple more questions. Do you want to read them out, Emily?
Sure. So we have a couple, and we only have another minute or so. So maybe another quick one. Could you speak a little bit about alectinib dose lowering? Some of our group members have had to decrease their alectinib dose because of side effects. Do you think that it can still be effective at a lower dose? And is that something that you do sometimes for your patients?
Was it alectinib, Emily? Was that the question?
Alectinib. Mm-hmm.
Yes, absolutely. So alectinib, with any of these TKIs, if you're having side effects where it's not reasonable to continue on the same dose, we would certainly take a little bit of a hold, pause, and then reduce the dose. And yes, alectinib can be effective at lower doses.
Okay.
Do you feel, sorry, go ahead, Emily.
Would you recommend, if a patient is on alectinib now, kind of preemptively switching to lorlatinib?
Again, I think that's a discussion. It's really, there's really no one right answer for every patient. I think if after a lot of discussion about the efficacy and the side effect data that you and your oncologist feel that that is the right way to go, then, you know, it's not, there's no one right or wrong answer.
Okay. Right. Well, we want to thank you very much for your time tonight. And if Mark can unmute everyone, this has kind of become a tradition for everyone to speak up and say thank you to our guests.
I saw the screen. I'll have that shit on it.
You
See,
guys
you
can
can
go
unmute
ahead and say
yourself.
thank you.
Thank you.
Appreciate you. Thank you.
Thank you.
Thank you.
Thank you so much.
If you could stay on too, there's a few more poll questions I want to ask.
Thank you very much.
Thank you. You guys are amazing. Good night. Thank you.
Thank you, doctors.
All right. I don't know if the doctors are still with us. But,
build out or like get someone to build out an office space in our basement.
Uh-oh, somebody might want to mute.
We might go back to the office.
I'm going to mute everybody, Mark.
yes. Okay. I think I'm the only one talking at this moment, but I have a couple more questions and I'm launching this poll. Where do you live? I'm assuming I will probably know the answer to this. Okay, 83%. Well, let me share the results. 83% are from the US, 10% from Canada, 1% from Europe, 1% from Asia, which is one person each, 4 from Australia. So I'm gonna go to my next question.
We're glad you're here, wherever you're from. Your family. You're our ALK family.
Okay, we've had a total of, whoop, let me launch this poll here. How many ALK talks have you attended live? So far this year, we have a total of 8. And I listed the ALK talks. So you can kind of, if you remember, or if you're in Lorlatinib, you might not remember.
or even Elective.
Yeah, that's true too. Okay,
The memory of a goldfish. They say the secret to happiness is forgetting, so we should all be good.
So we
I'm
have 4,
I'm
which is
extremely
embarrassing.
happy.
So it's about 50% at 23, and then we have 5 at 17, and 3 elected. So it's kind of scattered, but the majority of us have seen many of these, which is great. And then I think I have one more question here. Now this is, how many ALK talks have you watched by recordings? So there would be a total of 7. 0, you can also vote 0 that you watched by recordings. Okay, I'm gonna end this poll in a minute. There we go. So 0% is 51%, 1, 23%, then 2, 12%. By the way, you can go onto our YouTube channel and see all of them. So how to access the recordings through our YouTube channel. And
I will make a comment that these polls are a little misleading because our European audience are sound asleep at the moment.
True.
That's true.
And
Last question. Are you receiving ALK email reminders? If you are, which means that you're also receiving the newsletters. If you're not, you need to go to ALKpositive.org and give us your email address. So I'm ending this poll and showing the results. Do you guys see that?
Yep.
Okay, so we got 88% are receiving the emails and 8% are not. So, or 4% don't know. So those are my questions, and I'm going to keep the channel open for all us who wants to stay. I don't know if Emily, if you wanted to stay, I don't know.
Yeah,
Are you available to stay?
Please stay.
I'm sorry
Okay.
that I had to cut it off somewhat abruptly, but we wanted to respect the doctor's time. They both really needed to get going by 8 o'clock. Next week, we'll have Dr. Gaynor on, and he's another medical oncologist and researcher. So some of these questions, hopefully we could ask him if you didn't have your question answered
Can
tonight.
you give us a preview of what we might see next week?
I think he'll probably talk about the research that he's been doing. He was one of our ALK Positive grant recipients in 2018. And so he has been looking at the immune system in ALK Positive cancer and trying to figure out what it is about the immune system in ALK cancer that makes it not respond to immunotherapy, and maybe what might help it to respond better to immunotherapy. Colin, is that what you think?
Yeah, that's about right. Sorry, I was just reading a question from Gina. Yeah, that's about right. Yep.
Can I ask a question in regards to Awad and Gaynor? Is there a big difference between their therapies or what they're researching?
They're totally different.
Mm-hmm.
I mean, they're both related to the immune system, but that's about as far as the commonality goes.
Yeah, the goal, Dr. Gaynor's kind of ultimate goal, I think, is to develop some kind of a cellular therapy for ALK cancer. So something kind of like a CAR T cell, but it would be a little bit different because CAR T cells only work against cancer proteins that are on the surface of cancer cells, and ALK is only inside of tumor cells. So this would be a T cell therapy that would attack, getting T cells to attack ALK cancer is kind of the end goal of his research.
I just want to share my screen real quick. You guys see that?
Yep.
So this is our calendar for the month of March. Just want to let you know our art therapy on Tuesdays. Now we extended one at night at 7 o'clock Eastern time, and then guided imagery on the 11th. Excuse me, on the 12th. Oh no, that's the 11th, 8 o'clock Eastern time. So, and also we've added happy hour on a cancer calendar. We actually do have happy hour. So, and that's on the 26th, and baking, obviously. So this I constantly post on the internet, and it's under the event section also, the calendar. So, and then on the 28th, in my opinion, is probably the most important meeting of all the ALK talks. And that's when our Facebook administrators and Gina and Kirk Smith are going to give us a little, background of the websites and the services that we offer. And there's so much that you can find on the website and on the Facebook that I don't think people really realize. So I don't know if Gina, if you want to say a word or two about that.
Yeah, we're excited about it. It's ALK 101, and we're really excited. The founder of the Facebook page is Tom Carroll, and he's going to join us and kind of give us a little bit of a history. So if you are a long-term ALK, you probably remember Tom. His wife, Marita, was a nurse and passed away in April of 2018. And so he kind of stepped away from the group a little bit, and it's now moderated by some other people, and they're going to help us also. They're supposed to be coming to ALK 101 to kind of show you around the Facebook page, and then Kirk and I will kind of show you around the website and let you know of all the awesome things ALK Positive Incorporated is doing. And so we're really excited, and we hope everybody will join us and really just kind of get to know the history of the group and where we're headed and what we hope to do. So we're hoping for really big things. So hope you guys will join us on March 28th. Is that right, Mark? Make sure I don't mess up the date. My Lord.
Yes, I need the calendar in front of me, but I think you're right. So
March 28th.
yeah, and I think if you want to raise your hand, I can call on you, and you can unmute yourself if you have a question or comment or anything.
I'll just let everyone know that Christina Weber posted into the chat the web address for the YouTube channel.
Thanks, Christina. I was thinking I could do it, but I can't like do it under pressure. I have to like have it all prepared or I would get nervous.
Okay, well, I guess no one has
Christina
any
is our
comments.
awesome secretary on the board. So everybody volunteers their time, but she's our secretary and she is absolutely incredible. So if you ever get a chance to thank Christina Weber, she's really helpful. We have lots of people working hard on the board.
But Christina's the boss.
She's the boss, that is for sure.
Can I ask you guys a question since I'm not on the board? Is we have a lot of committees that we need to fill positions with, and we need more help from our members. And right now we have 73 members online right now. So can you give us, Gina, since you're our president, give us an idea of where could we find the committees that might be listed? Is that something you'll be going over on the 28th?
Sure. Yeah, absolutely. We're going to go over that on the 28th, but I can kind of go over it a little bit right now. We have our fundraising committee, which is an incredible committee headed by Misha Garrison. Her husband is ALK positive, and we have the medical committee, which is a very important committee headed by our very own Colin Barton. And Colin, would you like to talk about the medical committee?
Yeah, I mean, the medical committee is kind of doing a lot of different projects, but our number one overall thing so far has been the whole process of putting out the request for research proposal applications and then sorting through those applications and choosing which projects we actually select. So that's been the biggest part of what we've done so far. But we've got some new projects on the horizon that we're just starting to get underway, looking how to connect biotech companies with venture capital, and looking to start a research newsletter update section. So we've got some new projects underway as well. So I'll let Gina continue on. And
Gina, if there are the committee heads on the Zoom, if you ask them to unmute, maybe they can introduce themselves.
There are some other ones here. I did want to just kind of go back to Colin, and the research and review panel is just an amazing portion of the medical committee. That it has a lot of processes, but one of the things that really makes me confident in fundraising, and if you've seen, we've like put on golf tournaments, and my son does Kicking for Cancer and lemonade stands, and all kinds of stuff. But the reason that I feel so confident and putting all my time and energy into that is because of Colin's research and review panel. They work really, really hard to make sure we're funding the most impactful research. And not only is it just the people that are on the research and review panel, they're using the key opinion leaders in ALK. So the top gurus in ALK are helping us to make sure that we're picking the best research. So that's one of the things that kind of makes me excited about, fundraising for ALK positive research. We also have Amanda Nerstad on here, and she is our ALK positive summit committee. And so our summit is coming up, and I can't wait for you to tell all about that, Amanda.
Yeah, our ALK summit, I think Kathy just asked, our ALK summit 2021 will be July 31st and August 1st of 2021. And it was a long discussion, and it will be virtual this year, but we are still going to do some amazing things that will keep everyone hyped up and excited to stay together. And our theme is Stronger Together. I will be putting out a post soon about it, but you are the first to know.
Yay! And we also have our communications committee, which works on all the social media to kind of get the news out. Kirk, it was on here. Is Kirk still on here? I don't know if he's still on here, but if you want to help on the communication committee, please let us know. We also have our legal committee. So anyone who has that as an expertise, we have a lot of contracts and things that we need looking through. And so it's headed by our own Michael. Oh my gosh, my brain just went crazy. McCarty. I'm so sorry. Michael McCarty. And so if you are an attorney or have legal expertise, then you could help him out on the legal committee. He has a few attorneys. ALK positive cancer doesn't really discriminate. And so we know that a lot of people who have a lot of talents get this disease. And so we can use you as a volunteer if it's something you're interested in.
Can I jump in for a second?
Sure.
There was something I think you told me new about helping people out with getting some finances together for medications. So I don't know if is that under his committee or.
That is the advocacy committee, and that is headed by Clark Evans. And I don't think Clark is on anymore, but if you are, Clark, hop in. But Clark Evans does the advocacy committee, and that's one of the things that he's looking for to put on the website. Basically, financial assistance for each type of medication that's offered, from Genentech, where we have patient copay assistance, or financial assistance from other places like McKesson and things like that. But it's really just a page of resources for you to be able to access if you need help with copays or anything like that. So Clark will be putting that on the website soon. And also Clark works with political advocacy. So if political advocacy is something that you're interested in, just send us a message at info@alkpositive.org and we can get you hooked up in the advocacy committee. We also work on medical advocacy. So that's just making sure that, people are getting the right treatments and things like that. You may not know that, but as a member of this group, you also are eligible for the second opinion program, which gives you a free second opinion if your insurance doesn't pay for one. And Colin has a lot to do with that too. So Colin, I'm going to hand that over to you if you'd like to talk about that.
Sure. So yeah, the second opinion program was started about 2.5 years ago. And one of the wonderful members came to me and asked whether there was any way that he could help patients that just weren't getting very good opinions. There's a lot of oncologists that don't really have the first idea about ALK. And so we kind of started up a program, and we've now had about 50 members have gotten a second opinion, or been approved for a second opinion anyway. Not all of them have actually gone through and gotten it done. But what happens is that we've got 4 institutions: Colorado University, NGH, and Vanderbilt, and John Hopkins, and actually UCSF. So five institutions. And you can get a second opinion at no charge if you need the financial help. And so normally that second opinion would run about $800 to $1,000. And that's being paid for through the program. And we've just got a very simple application process. You just fill out a form, submit it. It used to be to me, and now we've got another member that you submit it to. It's all on that form. And if you've got any questions, just send a question, or you do a search on the Facebook group. Probably first you find all the information there actually. But if you have any questions, send a question to info@alkpositive.org and we can send you in the right direction.
One of the things that I saw when I first joined the Facebook group as a nurse is I just didn't realize that there wasn't a standard of care throughout the United States, or even throughout the world for that matter. And it seemed like, if you weren't being treated at a major medical institution, that impacted the quality of care that you got. So it seemed that where you lived a lot of times determined if you lived. And so this second opinion program was just one of the most amazing things that was brought up, so that people could actually have an ALK expert, consult on their medical case. And so I think that it's one of the most amazing things about being a member of this group. There's one more thing I wanted to see. Colin, would you like to talk about the survey at all?
I can give a teaser. So we've been working for quite a while now. I've put in more hours than I want to think about. We've created a very, very, very in-depth survey that should be getting released in the next month to 2 months max, we hope. and it's going to be HIPAA compliant, which means all the confidentiality things are all set up. It's done through a outside company based in California that does medical records and medical surveys and things like that. And the survey's basically going to be, the goal of it is to try to provide information to everyone that can help us. So advocacy organizations, researchers, oncologists, and it's going to provide them information about treatments we've been on, biopsy results. It's going to provide information about what our diets and physical activity levels have been. It's going to provide information about where you've lived, and they're going to try to, we're going to get a whole bunch of people to try to analyze the data and come up with things that can result in better treatments or better outcomes for us in the future. So look forward to a big release, press release about that when we get it going. And I do ask everyone to not only participate, but more importantly, to share the link to it with your local oncologist and encourage them to share it with other ALK patients who may not be members of this group, because this is not just for ALK positive group members. This is for any ALK positive patient, whether they're a member of this group or not. And that data is going to help all of us. So I do ask you to all get really involved with that once we get it released. Thanks.
That was one of the things I didn't know. Like, I thought if we just figured out, like, which sequence to do drugs in, and doctors just talk to each other, and then you could just change, clinical practice. But that's not the way it is. You actually have to have that data right there in writing, so, and research papers written on it before doctors actually change their practice. Even if we know that it's important to get an MRI, even if you're asymptomatic, until that data is there and in writing, it can't really be practiced, and it can't be, a standard of care. So I hope you guys will all participate in that. I wanted to highlight one next thing. It's Erica Lovack and the store for ALK Positive. She's the manager for it, and she's made all these cool shirts. So if you haven't seen this one says "More Research, More Life," and then Mark's shirt is the molecule for Lorlatinib, which is super cool. And so anyway, Erica, I'm going to hand it over to you to talk about the store. Oh, Mark, you have to unmute her. Can you unmute yourself, Erica? There you go.
Hi, everybody. I'm Erica Lovack. I was diagnosed in 2017 and had 2 years on Elective, and now I am on Lorlatinib. And I had hundreds of brain mets when I progressed on Elective. And in 3 weeks, Lorlatinib cleared the entire, I had a clear scan. So that's kind of my quick story. And I've been on Lorlatinib now for 15 months. So there's a lot of hope out there for anybody who's newly diagnosed. But I like apparel. And so I have my own nonprofit that helps fund lung cancer research. And we do a lot of apparel. So it was a natural fit for, I'm on the board at ALK Positive. And so I just started a store for us. A lot of members always wanted to get a shirt or get something. And we always had to do a custom order with custom ink, or we had nowhere to just have shirts all the time or apparel. So this was an opportunity for the charity to be able to offer it so people can get whatever they want whenever they want. We don't have to do a fundraiser to do it. So there's t-shirts, there's long shirts, there's hoodies, there's masks, there's bags. It's a way to support. A percentage of each sale goes back to the charity. And it's just kind of fun. We just started a research line like Gina has. There's a couple of options for research shirts. And then there's, I had a request this week to have Bergotinib also as a molecule choice. So now my designer designed the Bergotinib molecule. So there's Bergotinib, there's Lorlatinib, there's Elective. It's just kind of fun. It doesn't say the name of the drug on the shirt, but it's the, it is the molecule compound. So those are just kind of fun. And so, visit the store, buy something, you're helping research, and you got something cool to wear. And they're soft and comfortable. It's quality clothing. It's not like itchy and scratchy stuff. It's nice stuff.
It really is. Yeah. And Emily just put the link up. Thank you, Emily.
Thank
And
you,
there's
everyone.
one more committee. So you can see that it takes a lot of people to kind of make all of this happen. But the finance committee. And so I see, I hope Bill is available to talk to us. Are you there, Bill?
Hey, Gina. Hey, guys. How are you?
Hey, Bill.
Sorry, I can't have my video turned on. I can't figure that out this evening. How are you?
Good.
Good.
So Bill is the chair of our finance committee. And one of the things that's so important is for us to really be transparent about where your donations and where your fundraising goes. And so Bill is so awesome. If you haven't seen our financial report, this is like the first financial report that has ever like brought me to tears, because I can see like where things are going, and it just brought me so much hope, because I knew that our fundraising was going to stuff that could potentially save my life. So Bill, I'll go hand it to you.
Well, great, Gina. Thank you. Yeah, it's really important that, you know, we're an organization that started organically, and we're heading towards our own 501c3, which has been mentioned in the past. So it's really important that we have high, levels of integrity, accountability, and that we honor donors' wishes. So with that, we, at this point in time, 100% of the donations that we receive go to research. That's our mission. And it's very critical that our partnerships that we have right now with Longevity and Go2 honor that as well. So they are two stand-up organizations that, like I said, support us in terms of not only the fundraising platforms, but also guidance as to how we conduct the research, vet the research, and even work with Colin's RRP team that we've talked about earlier tonight. So we are in good hands, and I think our donors are in good hands. And when we become a C3, we will be a stand-up organization with high credibility with Navigator and all that. We're gonna be investing in more research, and that's where we're gonna go.
Awesome, Bill. And it is so important. And I'm going to kind of make this real, but this trial that we just learned about tonight, on Tuesday, I have my PET scan, and on Wednesday, I have my MRI, and it's going to determine if my brain mets have grown. And if they haven't, then I'm going to be eligible actually for this SHIP2 trial that we just learned about. And so it's so exciting to me to see that, all of our effort and working together as a patient group is really doing something and making an impact for the future of ALK positive patients. And so I don't know if I'm going to be eligible or not. I'll let you know on the Facebook page, just kind of the progress of everything that's going, but it's pretty scary when you progress off Lorlatinib. And so knowing that there might be other options, they wouldn't have been available if you guys hadn't all stepped up to the plate and had lemonade stands and donated. And so thank you so much.
I will just say with phase 1 clinical trials, people are like, well, there's only 10 people can get into that, but realize that's how Elective started. That's how Lorlatinib started. These all started with firstly benchside research, preclinical research, and then phase 1 trials, and then phase 2 trials, and then phase 3 trials. And we're all here because of those trials. So don't think it's just, only 10 people can get on those trials. This is what leads to the future.